We have finally arrived at energy reactions in the cell, part 5. This is the final chapter, and this deals with something called fermentation. So in this short video, you're going to have to be able to define fermentation and distinguish between the two types of fermentation, alcoholic and lactic acid fermentation. Also, we're going to wrap up some few things with how energy is stored. So we need to talk about calories and what a calorie is. And you'll have to identify some common compounds used for energy storage in various living things. I know this equation looks familiar, and we've been dealing with cellular respiration and breakdown of glucose to get energy in the form of ATP. But we've been concentrating on what happens with oxygen and when oxygen is present. Well, we all know that when oxygen is present, we can go ahead and get a whole lot of ATP energy. Thus, if we're following aerobic pathways, we can take our glucose molecule and through glycolysis, break it apart, end up with some pyruvate, and we do indeed get 2 ATPs out of it. And we were saying that that's not enough to keep complex multicellular organisms alive for very long. But if there's oxygen around, if we can follow these aerobic pathways here, we can go through this rest of the process of respiration where we can get up to 30 ATPs of energy, and that definitely is enough to keep complex multicellular organisms alive. But what happens if oxygen is not present and we have to go ahead and follow some anaerobic pathways? Here's where fermentation comes into play. We still go through glycolysis. We still get our 2 ATPs worth of energy, but now we have to follow an anaerobic pathway without oxygen. And as it turns out, we don't get any more energy from that. We're still stuck with just having 2 ATPs, but it does allow us to avoid some other problems. We can use this as our working definition of fermentation, process by which organisms release energy from food without the use of oxygen. But if we use that definition, we're talking about glycolysis being a part of it, because glycolysis is where we're actually getting the energy. The rest of the fermentation pathways, regardless of which type of fermentation you're talking about, really doesn't release any energy. It just takes the place of pyruvate conversion, citric acid, and electron transport, the rest of cellular respiration. Okay, it turns out there's 2 types of fermentation. The first one is alcoholic fermentation, done by several unicellular organisms and others, where after glycolysis, the pyruvic acid is going to be broken down into ethanol and some carbon dioxide. Ethanol is another term for ethyl alcohol. It's the same alcohol that people would use as drinking alcohol. So certainly distilleries and breweries make use of this process. Most alcohols are indeed toxic. Ethanol is no exception, and certainly if you drink enough ethanol, you're going to poison yourself. The carbon dioxide that is released in the process is oftentimes just lost as a waste. Sometimes distillers or brewers will trap the carbon dioxide. So if we're looking at sparkling wines, champagnes, stuff like that, the carbon dioxide that's in those products are actually the waste products of the fermentation process. By the way, you're never going to be asked to reproduce this particular equation. Let me just leave it here for completeness sake. Don't worry about it. We're more concerned with the just bottom line definitions of what happens in fermentation. Okay, with alcoholic fermentation processes, we're going to start out just like we do with glycolysis. And I've summarized a whole bunch of steps here, but remember, glycolysis wasn't too detailed to begin with. We pump in some ATPs so we can go ahead and split off the glucose molecule into two smaller parts. When we do, ATP energy is released, so we've got our net profit of 2 ATPs. That's kind of cool. We still end up with 2 pyruvates, and in the process, a couple of hydrogens are stripped off, and those are carried away by the NADHs. Well, this is all well and good, except pyruvate tends to be toxic in itself. Plus, if all we're doing is constantly removing NADs, we're going to end up running out of NADs here. So we need some ways to recycle this. Well, in fermentation reactions, we're going to go ahead and take our NADH and, through a series of reactions, make the hydrogen leave the NAD and latch on to what was our pyruvate compound. So give it back its hydrogen, but then strip off a CO2. And of course this happens twice because you got 2 pyruvates. And this will allow us to recharge our NADs, so we're never going to run out. We do end up with CO2 as waste, and then ethanol as a waste. And while it's true that ethanol is going to be toxic, it's not usually as toxic as pyruvate to cells, so we do have some time to process this and remove it, at least if it doesn't accumulate too quickly. Right, due to the potentially toxic and damaging effects of alcoholic fermentation, animals tend not to make use of alcoholic fermentation pathways. If they did, that would mean that any time you pursued any type of anaerobic activity, like all-out sprinting where you're going into oxygen debt, and again, pursuing anaerobic methods of processing energy, you'd be producing ethanol. And eventually, not only would you end up being tired because you're not producing enough energy, you'd end up, in essence, falling down drunk as well. So probably not a real good idea. Instead, when faced with anaerobic situations for any length of time, animals tend to pursue lactic acid fermentation. So here, we're going to undergo glycolysis just like before, but the pyruvic acid is going to be broken down into something called lactic acid, less toxic than either pyruvate or ethanol, and although it itself will have some undesirable effects, it will eventually allow you to kind of sort of keep going rather than conk out entirely. Lactic acid fermentations follow the same first step of glycolysis, so we're still looking at that glucose being broken down into pyruvate, and we still end up with the same problems of running out of NADs. So we have to recycle the NADHs back into the process. But again, we're going to have a different set of reactions here. And instead of giving the hydrogen back, stripping off carbon dioxide and getting ethanol, we're going to go ahead and just give the hydrogens back, rearrange all the bonds that make a pyruvate, and end up with a compound called lactic acid. Do you have to know the formulas for this? Do you have to know the summary equations? Nah, not at all. Again, just bottom line. What you do have to remember, though, is that the only energy that you're getting from fermentation pathways is the ATPs from glycolysis. And remember, since you invested 2 ATPs and got 4 out, you really only have a net profit of 2 ATPs. So anaerobic pathways aren't necessarily going to keep you going for very long. So we have to have some way of maybe storing energy and kind of sort of making it last. So let's take a look at some of those methods. Certainly, if you look at any food that you eat, there's going to be stored energy in the food. Not that this is wholesome, nutritious food, but certainly there's some energy here. So if you look at, say, the nutritional labeling, you'll find all kinds of information in terms of what is there for you in terms of nutrients, but there's always going to be something there in terms of how much energy it supplies you with. And here you're looking for calories, right? Calories give us an indication of how much energy is going to be stored in a particular food item. Now, a chemist or a physicist use the term calorie with a small c, and that unit is the amount of heat energy that is required to raise the temperature of 1 gram of water by 1 degree centigrade. Now, this is a very, very small unit of energy. So nutritionists oftentimes use calorie with a big C, or kilocalorie. A kilocalorie, calorie with a big C, is the amount of heat energy required to raise the temperature of 1,000 grams of water, or 1 liter of water, by 1 degree centigrade. And you can actually measure the calories in a food object by doing just that. There's a device called a calorimeter, and you could actually go ahead and take your food sample, put it in this little test jar here, this little test cell, and find some way of burning it and measuring the amount of heat that's given off. So if you wanted to actually measure the calories of food, this is one crude way of doing it. Now, how many calories do you actually need to keep yourself alive? Well, it's going to depend upon a lot of things. Your age, certainly your activity levels. The more active you are, the more calories you're going to need. So, generally speaking, if you just want an average ballpark, it's somewhere around 2,000 calories per person per day. So eventually living things are going to end up with a lot of glucose, either glucose that they have made from photosynthesis if you're a plant, or glucose that has accumulated through various digestive processes if you're an animal. If you're a plant, you're going to store excess glucose in the form of starches or maybe even simpler sugars. Think of the sugars that might be in sweet fruits. Think of the starches that might be in things like a potato, right? But you can also store it in liquid form, or plants can store it in liquid form in the form of oils. Think of corn oil, canola oil, olive oil, all the different things that people cook with. If you're an animal, well, you're going to be relying on a certain amount of glucose in your body fluids, in sugar, blood sugar. Matter of fact, most of the time you are operating off of blood sugar. Well, you can only have so much blood sugar. That's when you start to get hungry, or one reason you start to get hungry is blood sugar levels start to plummet, and the body is telling you that, hey, I need some more food. Well, you can store glucose in a compound called glycogen, and this can be packed around muscle tissues or in the liver, and can be converted directly to glucose in a pinch. For vigorous activity, you have enough blood sugar to last you an hour and a half to 2 hours, depending upon how vigorous. After that, you rely on glycogen reserves. Well, if you run out or low on glycogen, then you'll start to attack fat reserves. So that's the body's ultimate form of storage. And Certainly if you run out of fat reserves or run low on fat reserves, then you're starting to be in starvation mode, and the body will go ahead and start using structural components for energy. And that's what starvation is all about, when you actually literally start digesting things like muscle tissue to go ahead and keep your body alive. Well, fairly short video, fairly short quiz, but there are some important things that you'll need to know for the exam and any other quiz you have in class. So be able to answer these questions. Matter of fact, please answer them on a piece of paper. Turn these in and be prepared for similar questions on an exam or quiz in class. Good luck and have a great afternoon, evening, or whatever the heck time you watch this.